If in the last episode we talked about amino acids and the blocks for deconstruction, now we're going to talk about the whole building, the proteins. Hi everybody, welcome back to the Premed Journal. I'm Maria, your host, and in this episode we're going to talk about amino acids. So, this episode is going to have 4 parts. We're going to talk about the peptidic bond, the structural levels, the function of the proteins, and the classification. We're going to start with the peptidic bond. What is the peptidic bond? Well, the peptide bond is basically when 2 amino acids join each other via a bond between an amino group and a carboxyl group. Okay, so very important. Here, the amino group always has to be first. So if you see a peptide bond, you will read it from left to right, and the amino group always needs to be first, right? So it goes something like this: amino group, carboxyl group, click clack, peptidic bond. Okay, then amino group, carboxyl group, click clack, amino bond, peptidic bond. So when we have more peptidic bonds formed by more amino acids, we're going to have a peptide, right? And here we have a dipeptide, which are 2 amino acids that do click clack. Then we have oligopeptide, which are less than 10 peptides forming these bonds. We have polypeptide, which are between 10 to 100, and then we have proteins that are more than 100 peptidic bonds. 2. Structural levels. A protein is formed by 4 levels: primary structure, secondary structure, tertiary structure, and quaternary structure. For the primary structure, we have the linear sequence of amino acids. Here we have a protein called chaperone that helps the sequence of proteins form, but it's not part of it, okay? The secondary structure is defined by local folding, and here we have the alpha helix that is a spiral shape stabilized by hydrogen bonds, and also the beta sheet. So is a zigzag shape, then can be parallel, which is it goes like this, or anti-parallel, which goes something like this. Okay, I'm gonna put a photo. And we also have a collagen helix that is a very strong triple helix that needs to have two amino acids. It needs to have a proline and a glycine, and the third one can be whatever, okay? The tertiary structure is the 3D final shape, okay? There are some proteins that stop here and don't form the quaternary structure, and some that form it, okay? So we have globular proteins that are soluble in water, because remember we have some hydrophilic amino acids, so this is what the globular protein has. And it has a metabolic structure. And we have fibrous protein that is insoluble because it has hydrophobic amino acid, and it works as a structural protein, okay? It has a structural function. Okay, so the quaternary structure are multiple protomers. Protomers are basically polypeptides, like what we talked about, the tertiary structure. These are protomers that come together to form the quaternary structure. Okay, so now we're going to talk about properties and function. Proteins have a solubility characteristic. Why? Remember we talked about hydrophilic amino acids. Okay, so they form an outside layer, they come on the outside. So because they're hydrophilic and they like water, water molecules get stick to them, and it gets form a solvation layer, which is basically a H2O layer. Then we have denaturation. This is when a protein under extreme heat or agitation or acidic activity, it gets decomposed and loses its properties and the other structures, except the primary structure. So the primary structure remains, and it cannot form back into a protein with a quaternary or tertiary structure. It stays like this, okay? But to not get confused on when it gets congelated, okay? Do not confuse it when there is lower temperature, so when, for example, a protein is freezed. Because when a protein is freezed, the electrons cannot work and they're in like standby. But if it gets to like a normal temperature, then the protein can work. But if it's extreme heat, then it gets distracted and the protein cannot work anymore, okay? So we have specificity, which basically means that each individual has different proteins that are coded by their DNA. This is why we're all different. And also, this is why organ rejection in hospitals and surgery happens. And here I'm gonna read to you the functions because there are a lot. So, it has a structural function, a metabolic function, a transport function, a contractile function, enzymatic function, hormonal function, defense functions, and reserve function. These are all the functions that proteins have. And the fourth part of this video, the classification, here is a easier thing, but we have basically holoproteins, which basically means protein plus protein, and we have the heteroproteins, which means and then we have the heteroprotein, which are basically protein plus a non-protein part, which we call a prosthetic group. For example, glycoproteins, lipoproteins, nucleoproteins, etc. Okay, so this was the video about proteins. If you would like to talk about another topics that I am informed about or that I study about, I will do them. Please leave me a review if you understood what I said, if I need to better something or anything. This was the video. Bye!